even had their did his PhD in the National Center of Oncology. Then he joined the National Center of Hematology in Moscow, and that's where he developed his interest in stem cell development, hematopoietic stem cell development. And those of you not familiar with that, this is very important, because hematopoietic stem cells, without hematopoietic stem cells, you would sit here only for a very short time. And after that, you would be anemic and have all sort of other problems and disappear. So that system is important. It's very interesting. And in addition, it's a very fertile area, of course, if you think in the future, for replacing hematopoietic effective systems. But for the moment, you want to understand where they come from and where they go to. And so Alexander subsequently moved from Moscow to Britain, and a short time to Rotterdam even. I always mention that if people even if they spend only there a week, I will always say, and he did his important discoveries in Rotterdam. But nevertheless, there, together with some colleagues, he found a very original place where initially these stem cells reside, from where they expand and form the hematopoietic system. Subsequently, he moved to Edinburgh, where he is now leading the Institute for Stem Cell Research, and he became there in 2006, was the professor of hematopoietic stem cell biology. So we look forward to hear how dependent we are from those cells.
Okay, thank you very much,
Anton.
Yes. Thank you for this introduction and inviting me here as well to visit Scotland. Right, so what we are working on, what my group is working on, is the development of hematopoietic stem cells. And just to introduce you to what hematopoietic stem cells are, they lie at the foundation of the entire hematopoietic system. And you can see that here it's shown that they self-renew, but at the same time they can differentiate and give rise to all cell types here, mature cell types, lymphoid and myeloid cells. So what is important is that also there is a practical importance for these cells, because when you do bone marrow transplantations to patients, to leukemic patients who underwent myeloablation, who were, for example, irradiated or were treated with chemicals to ablate leukemic cells, then normally they are transplanted with bone marrow from donors to reconstitute their normal hematopoiesis, because in those treatments the normal hematopoiesis is also affected. Just to introduce several important assays for three types of cells in the hierarchy, which I will mention during my talk. Hematopoietic stem cells, you really have to do transplantations and look for a relatively long time into the irradiated recipient, look in the bone marrow, spleen, and thymus, and detect by flow cytometry the presence of donor-derived cells. And if after this long period of time they are there, then it means that you transplanted true hematopoietic stem cells. And that's because it can self-renew. That's why it can sustain, can persist in the hematopoietic system of recipients for a very long time. And here you can distinguish this from the remaining host cells. These cells give rise to splenic colony-forming units. This is a much shorter assay. After 1 or 2 weeks, you find colonies of blood cells or myeloid cells in spleens of recipient mice, and each such colony is generated by a single progenitor cell. So what you can see, they are more mature than hematopoietic stem cells, They have some capacity to reproduce themselves, but it's very limited. And then you have colony-forming units, which are downstream of the hierarchy, and you can test them in the methyl cellulose. They form these colonies, and they don't have self-renewal capacity. So just to tell you about our direct interest and how we contributed into the field. So, and just to set the background, really, I think, which is important. So that was, for a very long time, it was suggested that the yolk sac in the very early embryos, so this is the day 7.5 embryo, and this is the yolk sac here. And you can see this red belt of cells surrounding the yolk sac, which produces erythroid cells. And this is the first place where you can find active hematopoietic events in the embryo. So it was suggested that the yolk sac gives rise to the entire hematopoietic system in the adult animal. And that happens this way. So you have the hierarchy, which has to travel to the fetal liver first, which is the main place of hematopoiesis before the bone marrow develops in the limbs. And then what happens is that in the adult mouse, around the birth, or slightly before the birth, stem cells migrate to the mouse. And that was based mainly on this research of these two eminent biologists who really did a lot in experimental hematology. What they did, they looked at the presence of CFUS in different tissues of the developing embryo. So this is the age of the embryo, and these are different tissues here shown. The yolk sac, the circulation, and the liver. And what they found is that day 8, so this is day 7.5 embryo, so day 8, they found that there are CFUS, splenic colony-forming cells in the yolk sac. And then they start seeing them in the circulation, and then they start seeing them in the fetal liver. So it sounds very logical, isn't it, that the yolk sac generate the entire hematopoietic system. So when we started to work on that, we found that actually that doesn't reflect a true situation. So what we found is that if you really radiate your mice well, recipient mice well, then they don't develop endogenous CFUS, because if you under-radiate them, so the mice tend to develop in their spleens these colonies, and so you can take them for donor-derived ones. So if you do good irradiation and do transplantations of early embryonic cells, so we did very careful analysis, you can see this day 8 embryo, we couldn't get any CFUS, any colonies in the spleen. And so we couldn't understand how it could be that you have red blood cells in the yolk sac, and yet you don't have hematopoietic stem cells. So you have kind of terminal or differentiated cells, but where are the founders of this system? And so the idea was to look into the body of the embryo, and we found that you have approximately at the same time the CFUS appear both in the yolk sac and in the body of the embryo. But what's interesting, the number of CFUS is higher in the body of the embryo. So these are somite pairs of the developing embryos, and you can see these are days of development. And then we did dissections of the embryos, and every time you have to do transplantations and look into spleens of irradiated mice. And we found that here you have, we identified the area around the dorsal aorta, which also includes urogenital ridges. So this is genital ridges and the mesonephros, which is enriched for CFUS, for splenic colonies. And this is on day 10.5. That's the picture, semi-quantitatively reflecting the situation. And day 11.5, what you see is that most of CFUs are concentrated now in the fetal liver. So, and at that point, we understood that was the real situation, and it's clear now that we were right. So not the previously published paper, classical paper, but this one. And since that, there are lots of things happen, and there is a very strong confirmation that true adult hematopoiesis originates in the area of the dorsal aorta. And there were recent publications about zebrafish where you can visualize cells, how they undergo this formation in the dorsal aorta and make a pool of hematopoietic cells later on. So that's the situation, but I'll just tell you that there is a little bit, I already mentioned that it's a little bit paradoxical that you have still cells in the yolk sac. So this is day 8.5 embryo, this is the body of the embryo. So the embryo is, you can see it's bent over the, its back here, and the body is really not formed properly. And this is the yolk sac here. So this would be, here would be blood islands if that would be not a fixed embryo, but, so you might be able to see it. So if you do transplantations, as I mentioned to you, you don't have reconstitution of the hematopoietic system in this mice. But if you take day 11.5 embryo and you isolate the area which surrounds the dorsal aorta, which is actually called the AGM region, aortic gonad mesonephros region, and that's how it looks like. This is the dorsal aorta, and these are urogenital ridges. And you do transplantations into this